Professor Idan Segev, who is going to talk about technology, new technology, and the future of the brain. I'm sure all of you have heard Idan before, so we're up for experience. Thank you, Idan. We might be interrupted in the middle when the Minister of Health of Greece is going to honor us with coming, but we'll see how it goes.
Okay, let's start with the science. Good to see full house, small full house. And so I gave this talk a bombastic name, the future of the brain and the future of technology. And so I want to convince you that there are some dramatic things happening in terms of technology that will have an impact on how we look at the brain, how we can research the brain. So I want you to become amazed from this talk, but I also want you to ask questions. So if somebody, because this is eventually the whole thing is about interacting with you, and it's not about me talking about what I already know, but trying to make it easy for you to understand. So please really don't hesitate to ask questions if something is not clear, because if I lose you, then I'm unhappy, and you don't want me to be unhappy. But before starting, I want to remember or to relate to Viviana, who you all know, I'm sure she was a very special, intense. I thought about the name Viviana because of Viv, vitality. She was a very intense woman, very interesting woman, and very close to us in many ways and very helpful. And she created this brain circle Italy following our concept, and was very instrumental and very generous also for ELSEC. So I will miss her a lot. And I actually talked to her a few days before her unexpected sudden heart attack. So this is a place for separation. But she's always there. It's always there. You know, it's interesting that you just yesterday talked with Moriel and Freddy. There are a lot of things in your brain that you're not aware of. And suddenly somebody comes and boom, the whole experience is there. So it's there. So when I look at Viviana, many things that I don't want to share with you are happening now in my brain. Probably in your brain as well. So the plan of today is really to give you very fast thinking about AI, because one of the things that you will hear much more about this tomorrow, especially from the talk by Chaim Sopolinsky, but also others, about the introduction of AI into brain research, but also both ways. So brain can interact with AI and AI can interact with the brain, and it's a very fruitful interaction. And a big change of the way we look at the brain, at the data, and about thinking about the brain with all these large language models. And Chaim will talk a lot about this. A new species that is living near us, the creation of this AI. So I'm not going to go much about this, but just to show you how the new technology, the AI, is in many ways inspired by what we learned about the brain. It's not an independent technology, so to speak. It is inspired by the brain, and then it does its own development. But I really want to focus today about two aspects that I think are really highly interesting for me. So I'm also learning a lot about the brain. You know, I'm doing what I know to do, but there are many other things that are happening in the field. So today I want to introduce to you something that you just mentioned, was mentioned today by these two British students, the organoids, the small brains that are being developed in a dish. I'll talk about these small brains, because I think this is really a revolution in many ways. And I'll show you a little bit about what does it mean, this small brain, how do we develop, how do we make a small brain, and what kind of use you can do with it. So this is really the focus of the talk. I will also talk about my own study today, on my group, studying human neurons. You know, we know a lot about the brain from mouse. Mouse is an animal that we know very well, we can manipulate genetically and so on, so most of the work that is being done in brain research worldwide is really based on mouse brain, but recently we can do more and more with fresh human brain cells, and we can learn about whether these cells are similar or not similar, or the network is similar or not similar to the mouse brain. Did what we learn from the mouse is relevant or not, or how much is it relevant? Of course, I don't have the answers, but I will show you the beginning of really working with human brain, fresh human brain, at the level of cells, and the connection between cells. So this will be my focus. And if time will permit, I I will give you also something that I think are really game changers that science will contribute not only necessarily from the brain research, but from other aspects of research. So very briefly about AI, you will hear a lot, as I said, about this neuro-AI new field, connecting neuroscience to artificial intelligence. So biological intelligence, PI to AI, back and forth. I just want to show you that we studied a lot the brain, from the whole brain level, to the network level, to individual cells level, to individual connection between cells, the synapse. We know the signals in the brain. We know quite a lot about the brain. And this gave inspiration to build machines that take some of the principles that we took from here and implement them in what we call today AI. So for example, you see this rough sketch of what we call deep neural network, a system of neurons. This is artificial neuron, this is a real neuron, artificial neuron, one layer, another layer, another layer, another layer. And we use this technology or this representation in order to, for example, do large language models like ChatGPT that you know all. So this is the foundation that works underneath when you type a question to ChatGPT. Of course, it was trained before. The underlying mechanism is this neural network, artificial neural networks with connections between these artificial neuron synapses, all based, or much of it based on what we know about connections in the brain. So this brain study, after so many years, eventually came to implementation at the level that we have machines that can talk to us, that can remember, learn, and so forth, but it's all based on this kind of mathematics. I'm sure Chaim will say a few words about it. And taking this technology, the deep neural network, AI technology, we can use it backwards, so to speak, in order to understand the brain. Haggai will speak a lot about implanting electrodes in the brain, reading a lot of data from the brain. AI can help us analyze the data, find the secrets, so to speak, within the data. AI can help us solve things that the brain could not solve. I don't know if you are aware of it, but there was a problem in science which is called protein folding. How does protein fold in 3 dimensions in order to be effective? So medication, medicines are built on proteins, some of them at least, and we didn't know how to design medication because we didn't know how to fold the protein in a way that would become effective. But AlphaFold technology, using deep neural networks and other systems, enabled now automatically to fold proteins and generate a new generation of drugs. So this is the result of the brain inspiring deep neural network and AI. AI help us to develop drugs that will help us to repair the brain. So this is a nice ping pong crosstalk between the brain, technology, and technology helping us to better understand the brain back and forth. So I don't want to say much more about this, but this is a complete revolutionary approach. The AI and most of the big centers today in the world are going towards neuro-AI centers, institutes, and so on. But today I want to discuss with you, as I said, the organoids. So this term means that you build a little organoids, organs, small organs of speak, very small organs. Maybe you remember from Sweden, Idan Segev from the Brain Circle in Sweden, in Stockholm. Idan Segev is our person of organoids. He's not here. And he explains there what do you do when you develop an organoid. So let me go very briefly about this technology. So you take a cell, let's say a skin cell, on my finger now. I did this. I took many cells from my finger, really. I took many cells from my finger. I then put it into a dish. And you can reverse engineer these cells. You can make these 75 years old cells to become stem cells. You can reverse engineer genetically these cells into becoming fresh, young, multipotent stem cells. They don't know what will happen to them, these stem cells, but they are just starting to live. So this is a Nobel Prize from Japan about 15 years ago. The fellow who developed the technology to take an old cell and make it young, fresh, stem cells. Then you can take these stem cells individual stem cells and force them, so to speak, to grow into an organ. You can make them grow by certain manipulations to become. Is he coming? If the minister will come, then I'll stop. Maybe if he wants, then he will give his introduction, then I will talk to you about small brain.
Of Greece, a visionary politician, a good friend of Israel and the Jewish community, and a good friend.
Thank you very much. Is that okay that we continue for half an hour or so? Thank you. Thank you for coming. So you take this skin cell, you make it, you reverse engineer it genetically to become a stem cell, then you put it into a dish, and with certain manipulation, you can generate this organoid. This group of cells, you can grow them, they split, they grow, they split, and you can force them, so to speak, to become a brain, or a heart, or a kidney cells. And you take a certain period of time, you can see what happens to it after 14 days, 35 days, 80 days, and so forth. Eventually you get this organoid, and these two students can tell you more because they do it physically in the lab. I'm not doing it physically, it's not my field, but I'm telling you that this is a revolution because you have a small brain of a particular type of cells or organ, and you can manipulate it, you can study it, you can check medication on it, because it's outside. It's outside. There is a problem with this outside, we'll discuss it in a second, but this is the technology. So now when I'm saying organoid, I mean a group of cells that were born here, became young and then goes about 80 days, you can do it more, depending on the type of cells and where it comes from. So this is a small brain, let's say 1 million neurons, about 1 million neurons, some bigger, some smaller, depending on it. And these million neurons are a small network of neurons. If it's a small brain, they interact and they have signals, chemicals, like a brain. One thing that they don't have is the blood support, which you need in the brain in order to make this. So you have a support of energy from the outside, which is a limitation I will discuss in a second. So that's the brain organoid. But it could be heart organoid, and it could be kidney organoid, but in this case we're talking about a brain organoid. Small piece of a brain that came from my own cell. Okay? So that's what I just said. These stem cells are then grown into a small cluster, about 1 million neurons, called brain organoid. And you can see that this organoid, if you look deep inside using technologies, you stain the cell, you can really see nerve cells and the way they look and interaction between them, synapses and axons, and you can record signals. So it really looks like a small brain. Of course, it is not exactly like a brain, because it is only part of the cell, but in principle, Liora, shalom. Come. Liora is the daughter of Dan and the friend of the two students you have. But now I want to tell you about something that when I read it, I thought I was really shocked, so I want to shock you too. It is called the brain chimera. You know what is chimera? Chimera was a mythological animal that had 3 heads. And this is a drawing from Italy, from the 16th century. A chimera. I want to show you a chimera brain. A rat brain or a mouse brain with an organoid that came from human. So it's a rat brain with a human brain inside. This is how it looks. This is a rat brain. I will discuss it in a second further. So this is a whole rat brain, and this is a piece of a human brain that was inserted into the brain of this, in this case, a rat. And then you can ask interesting questions, also ethical questions, philosophical questions, about this new chimeric brain. It's a hybrid brain of a rat with a piece of human. You can ask whether these human cells, about 1 million human cells, will they interact with the rat brain? Will they create a network? Will it become human? What's going on? The advantage of this piece of brain that is inside the living rat brain is that you can check medication in vivo. Rather than in vitro outside, now you can really test whether the medication gets there, how does it affect the human neurons inside the rat brain. I thought this is an amazing jump which happened about 2 years or 3 years ago in Stanford, and I want to elaborate a little bit about this magical thing, you know, with all the questions that may arise. Okay, so you start with this organoid, you put it in the brain of this, in this case of a rat or a mouse, surgically, you just put it surgically inside the brain, and then you can test, for example, whether blood vessels, as I mentioned before, you don't have usually blood vessels in the organoid when it's outside, but when it is implanted into the brain, these blood vessels are starting to grow from the mouse blood vessels into the human organoid and start to support it energetically. So you don't need anymore to bring any additional support because the brain itself has this mechanism to make this group alive. You can also record with our techniques. You can put electrodes into this place and you can record electrical activity. So you see that these neurons, group of neurons, are really active. It's not that they are sitting there passive, but they are active, they generate signals, interact with each other and so on.
The animal blood is compatible with the human blood?
The what?
The animal blood is compatible?
Yes,
Yeah.
in terms of supporting energetic support. Yeah. That's the yeah. It's not only, I will show you in a second, it's not only the, because in the brain, okay, that's a good question. In the brain, the blood vessels are giving you energetic support through another cell that connects between the blood vessels and the nerve cells that are called astrocytes. So the blood vessel has the sucrose and the energy that enables the brain to generate all its activity, but we need to take this energy into the nerve cells. And there is this intermediate cell, which is called astrocyte. We'll talk a little bit about that. And if you remember from last year in Vienna, in Pal Goshen gave you a whole talk about astrocytes. I will say a word about astrocytes. So it's not only that this group of neurons are supported by the blood vessels, but also by the other cells. So they are integrated into the brain of the mouse after a certain period of time. It's not immediate. Yes.
The brain is still in the rat.
It is in the rat. Yeah, it's a full rat.
Does that change the behavior of the rat?
That's the question. Will the rat start to speak? Ask a question. No, this is a serious question. These are serious ethical questions. I'll discuss it in a second, but that's of course. Yeah, but it is in the living brain. It's a small portion. I will show in a second that one question that was recently asked is whether when you take the visual, you take visual cortex or part of the visual cortex of the rat and make it blind surgically, when you put the human organoids into the missing region, will the rats revive vision? And the answer is yes. So this is not only functional, it can bring function, part of the function. So that's a new technology, the dramatic technology, to take human brain into the mouse brain and use it for different purposes. And so I will show you the different purposes. Okay, just a brief, not to be too scientific. So this is the process of putting this organoid inside, waiting a little bit, and let the organoid grow and integrate. And then you can have some technology like this, using a lot to stain a region and to see whether this region is connected to other regions. Just stain and just see the fibers going from the implanted organoid to other regions and make connections and so forth. This is what I'm showing here. And you can see that the period that it takes, you have to remember something important, that human brain develops much slower than the mouse or rat brain. So if you put an organoid at this certain age of 80 days, it's a very young, it's a pre-born baby. Yes? It takes time for this system to grow into a mature circuit. So it's a very young human brain, so to speak, into an adult mouse. Because the pace of growing is different between human and mouse. So there are many issues there. Just wanted to show you about the fact that they become integrated and they grow, the organoids grow inside in volume, and they make connections. A lot of fibers are going between inside the organoids and also from the organoid this direction and from the mouse brain in this direction and all these other regions. So this is a way to see whether different parts of the brain, from this here, from this here, from this here, are connected, yes or no, to this new organoid. And eventually you see the electrical signals and you stimulate and you recover partially the visual system of the rat. So that's in some sense a way. Of course, you can ask yourself, why do you need human in order to recover the visual system of the rat? I can take an organoid from the rat and put it into the rat. Why do I need human? But in this case, it's just to show that some of the function also is recovered. Not only the activity, but the function, vision becomes better in the rat because you put it, a human brain organoid, into the visual cortex.
Is that going to lead to judgment? In other words, by integrating all these things into a rat's brain, will the rat one day be able to exert judgment?
Judgment, the seek judgment, is a judgment between
Means
good and
good
bad.
or bad?
Consciousness.
It's like Millie's
That's the question. How human is this rat, if at all? At what point will I say, okay, this rat has a human? But as I said, it's much too early. We are not yet at the period when we waited enough for these rats, and there are ethical questions. Do we want to do it? Yes. What will happen if the rat would start to be conscious and will know about its own existence? I don't know. There are many issues that you have to discuss before you allow this thing to develop. I'm just saying that this is just the beginning. It's a beginning of a new technology. And the main thing of this, the main purpose of this technology is really to test medicines in vivo. Something that is very hard to do in human. Yeah? I cannot go into human and check by electrodes or by some local recording into your brain whether this is functional or not functional. But here you can do it. So that's really the main, the declaration of the different teams in the world that are doing it.
Excuse me.
Yes.
Just to follow up that question, is there a point in times where the human neuron are functionally indifferentiated from the rat?
They're functionally
Undifferentiated from the
undifferentiated. When you
you
mean
cannot see the difference. I mean, of course, we always look at the DNA and say the
Mhm.
difference. But is there a point when we can say, okay, they just behave like
no, they remain at least for the several. I think it's one year now. At least for this 1 year of rat being 1 year with the human brain, it's still these nerve cells remain with properties of human nerve. They are bigger, they have more connections, and many things that are typical to humans. So the genome of the human brain remains the main characteristics, but there are connections, so they do interact, but they are very clear set. You can stain them in a different stain, so they remain different. I just want to tell you that we now can do, we scientists can do now what they call connectoids. You take one organoid, they take another, this is a Japanese, one organoid, another organoid, and they build a little pipe, a little road, so to speak, that allows the fibers to connect from one part to another part in order to understand better, connectivity within the brain regions. So this is another tool that is now being developed. I mean, these days it's very, very fresh. I learned a lot about this. And in last month, there was a new paper from the Stanford group who actually developed the original thing. They call it assembloids. So there is the organoids, there is the connectoids, and there is the assembloids, which is a group of organoids, but from different parts, or you develop them from human, but from different, or from mouse in this case, from different parts, or make them different parts of the nervous system. So you want, one thing that we don't easily understand, or difficult to understand, is how does stimulus, sensory stimulus from the periphery, goes into the spinal cord, through the spinal cord, into particular brain region, and eventually, let's say you see or you feel touch, and something happens here. So the whole stream of information from the sensory input through the spinal cord into the spinal stem cell and so forth, eventually to the brain, is not easily studied. So this group is now developing these assembloids where they take different parts or develop organoids from different parts of the brain, from the sensory system, from the spinal cord, from another regions, up until the cortex. They connect them together now, they connect them together in the dish in order to understand what happens when you stimulate the sensory organ here, how does information flow until this part responds, let's say, to vision or to touch. So I was quite amazed by all this. I hope that yes.
You said that the organoid is you grow the organoid before you implant it.
Yes.
Does it continue to grow after?
Yes. Yes. That's what I showed here. See here? You the organoid that you implanted, this is 1 month after the implantation of the organoid. You can see what happens after 2 months, after 3 months. So this is this graph. You see what happens after 3 months to the volume of the human organoid. So the human organoid split, grow, split, continues to grow inside the brain of the mouse. And continue to grow.
Is there a rivalry for space between the human cells and the mouse cells? Do they fight?
Yes, because there is a limitation of space, so you have to, decide who. It's good, and I will show in a second that yes, there is rivalry, but eventually the human wins over. Correct. The human cells, I mean, takes over in some cases. Okay. So last year you heard in Vienna in Bad Goshen, as I mentioned before, talking about this another cell type, which is about 50% of your brain. It's not only nerve cells, but what is astrocytes, or in general terms they call glia cells. These astrocytes have a very different shape compared to the neurons. I will show you here a video. So this red piece is part of a nerve cell. It's a process. It's called dendrite. And around it, there are these many of these astrocytes engulfing it, around it. And let's see that the movie will walk. Okay, so this, if you see all these little brown cells are mushy, the spongy cells are engulfing around, the dendritic tree here, and supporting it through a blood vessel that you don't see here in addition. So this kind of interaction between the blood vessel and these supportive cells. Inbal doesn't like us, and she's right to call it only supportive, because these cells are also functionally important. They are part of the memory system, and they not only support, but they play also into supporting and helping the interaction between neurons. So it's much more than a support, but you can see that this is a different type of nerve cells. And actually, when you look at them like that, stop the movie so your attention will go not to the movie, yes. So this is an example of another technology today that you can, in a second I will show you, you can take astrocytes and put them into the brain of a mouse or of a rat. Why do you want to do it? Because you want to ask a question whether this bigger, so human astrocytes are much bigger. So these are astrocytes, the red ones are astrocytes from mouse. The green one is an astrocyte of human. It is much more extended and branchy, and apparently much more effective in supporting energy. So the question is, if I will take a mouse and I will put astrocytes into the brain of the mouse, will the mouse become more clever? It's supporting energetically and solve things that the other mouse without human cells can. So this is another chimera, but in this case you don't take the whole organoid, but you put individual cells or a group of cells of a specific type into the brain of a mouse. So this is a chimera. You took these cells, you put it into the mouse, and these cells, in response to your question, these cells are taking over. They destroy, so to speak, the astrocytes of the mouse, and the whole mouse becomes covered by astrocytes of human and not by astrocytes, previous astrocytes of mouse. This is again a very recent work. So I'm learning. One good thing about coming to teach here, to speak here, is that I need to learn something new. So because I need to learn something new, I'm reading things that I would probably not read otherwise, because I need to read also what I'm doing. So once a year I'm reading things for you. That's an interaction. So you can see, without going into details, that in this red brain that is now fully covered almost, with astrocytes of human, the synaptic connection, which is measured by some kind of electrical activity, the synaptic connection, the red ones, and the capability of these rats to learn maze are much better than the previous. So this is a genius, an Einstein rat, that, so to speak, that became much clever in solving real-time problems like a maze, due to the fact that you implant it not with nerve cells, but with this other cell type, which is very important for energy support. I think it's quite amazing, something very new for me, without going into the details. If you have a question, I'm going to something else, my own work. Okay, chimeric brains, both using organoids and also using cell type in order to check what will happen. I can summarize what I said, that there are these many questions that are being asked using these organoids. For example, Rand Shorer is using the organoids in order not to study Alzheimer's necessarily, but another disease, but you can ask, you can take a cell from a grown-up person that is Alzheimer's person. You can take a cell from his own or her skin, reverse engineer it to become a stem cell, look at the organoid that will eventually develop aspects of Alzheimer's or another disease. You can check, you can look at the development of disease because you already took a cell from a person that is about to become Alzheimer's and so forth, and use this technology in order to study the progress of the disease, or maybe be able to stop it along the way and so forth. But that's a very important use of this organoids.
So in theory, nothing will stop us to be able to build spare parts, pretty much.
Spare parts.
If you have a brain, brain damage, you take skin cell, you grow your organoid, and you replace your own.
That's
brain.
the whole idea. That's another idea.
Can you have it
not only for drug development, but also for building spare parts, if you want to call it. Yeah, like a heart.
Yeah,
you take from your own skin, you develop your own heart, and then you implant with your own. Then there are all this issue of rejection and all these other things that you need when you take a tissue from another. So that's a direction.
Yeah.
To build parts.
Can you please hurry?
Sorry?
Can you please hurry?
Hurry?
Yeah. We need
I will
that
hurry.
soon.
When do you need it?
You don't know.
I will do my best. I mean, the group of scientists will do it. But there are, of course, many moral issues, and people are very careful, and maybe we'll have time to discuss it. I don't want to go about this regulation, what is allowed and not allowed, and so forth. If I will have time, and probably not, I will tell you about personal issue with the ethical question that I had with the Chinese fellow scientist that is doing something else, but I don't want to distract here. I want to remind you that from last year, my talk was about connectomics. Last year I showed you another technology, not new, but the technology that enables us now today to really take a big pieces, relatively big, of a cubic millimeter piece from human, from mouse, from monkey and from mouse, and reconstruct these pieces, reconstruct them. This is post-mortem, this is dead tissue, but in order to analyze the anatomical connectivity, cell types, and so forth, you use this connectomics project, which is actually being done in several places in the world. It's a very expensive project. You need many, many microscopes to cut, and then reconstruct the whole jungle and all the connectivity of this group of cells. So this is a new information that came to my lab, because now I have the information of connectivity and also the detailed information about a single cell, which you can really reconstruct today. A single cell, this is a human neuron from the human cortex. It's post-mortem, as I said. But now you can reconstruct, using this connectomic, the full neuron with all its beautiful. Can you turn off a little bit the light in front so you can see? Is it possible to turn on it there? No? No? Okay. Anyway, so this is a human neuron. You can see when you enlarge, you can see it very well, but when you enlarge it, there are many, many little branches, but synapses, about 10,000 synapses on this neuron. And you can see a mouse neuron also reconstructed using this technology. So this is human neuron, so you have much bigger neurons in your cortex compared to the mouse neuron. And you can zoom in and you can see all the connection, the synapses on a small branch here. So this is a beautiful new data, and so my 4 students became in the last year a connectomic group. They are working on this connectomic data to ask all the questions, but in new ways. So this is one aspect of the data, high resolution anatomy of human neurons and connectivity. The other data comes from operation, because you want these neurons to be alive, to function, to generate electrical activity, chemical activity. And for this data, you go to hospitals, including Shaarei Tzedek in Israel, and others in the world. And because physicians every day do operation on the brain, in this case it's a human man brain, you take a piece of a brain here, so it's a piece of a living tissue, yes, of the whole thing. So the cancer or the tumor was here, but this part of the brain seems to be fine. So So you can use, because you have to go through and take the whole piece out, you can use these pieces of a healthy part of the brain to study the physiology, the connectivity, activity of this piece of the brain. It's a living human brain, not outside now, because you have to take it outside anyway. So this was done many years before, but only in the recent 10 years, scientists and physicians started to collaborate on this type of tissue. Otherwise, the tissue would have gone with do some analysis of the type of cancer and then you throw it to garbage. But now we can use it for what I'm going to show you in a second.
If they need to take it out anyway, if you can put it back in the mouse brain, why can't you just put it back in the human brain? If you take out the tumor and then you put back the extra bit, that still works.
I must say that I didn't think about this idea. About this idea whether you can take, I mean, because as I said, this part of it is of course cancerous or tumor, so you have to take only the good part. Let's see. But by the way, I just wanted to show you, I'll come back to you, just to show you the size of the piece that is being taken out from the human brain relative to the whole brain. So this is the same scale. So this is about cubic centimeter or so, and this is the whole brain of the mouse. They typically take in operation a bigger brain than the whole brain of the mouse. So the question is, yes, there are parts of the brain, in this case this part, which is the cortex, which we feel is healthy, but you have to take it out. But your question is whether I can take part of it that I think is healthy and implant it back into the brain. And you remember there is now a big missing piece, so it's not easy for this part of the brain, even if I implant it, to grow into the tissue, because the tissue is not there. Most of the tissue is not there because it was doomed. But I never thought about nobody ever did it as much as I know, implanting that piece of the brain of maybe one human brain to another human brain. It's another chimera, so I don't know the answer to this. Anyway, we have now a big database of cells, in this case the anatomy of the cells, yes, but also recordings from individual cells, activity, chemical connection, synapses. So we know a lot, much more than we knew before. And we can ask questions. I will show you very briefly one question that we asked in the lab, and I don't want to go too further. So one question we can ask, and this is published from a big book, I was part of the modeling part of this book, is whether there are cells in the human brain that are genetically different, that are very different than the cells in the mouse. Are there new cell types? New cell type that does not exist in the mouse. Not necessarily it would be, but yes, apparently yes, we have new cell types, not more cells. We have more cells than the mouse, but also new cell types. We discovered few genetically new cell types that have a particular behavior, and we think that these particular cell types may be responsible for specific diseases that are unique to human, maybe schizophrenia. I will talk about schizophrenia. Okay? And you see also that the nerve cells, the individual cells in the human brain are much bigger than individual cells in the mouse. They are more separated, sparse, much bigger, more connections between cells. So we start to know more intimately human neurons relative to mouse neurons. And then we take, in our lab, in my lab, we take the anatomical data that I mentioned, the physiological data that we recorded from this living tissue in the dish. By the way, you can put it in the dish for months. And the piece of the human brain taken from the human brain can continue living in the dish, not growing, but living in the dish. You can record from it and analyze it and so for a long time. Then you take mathematical equations to try to understand how this nerve cell, which is our basic microchip, how does information flow from the connection of the synapse here to the output here? What kind of microchip is this cell? What does of input-output properties this cell has in the mouse versus the human? Or in a popular way, I would say, are human neurons more clever than the mouse neurons, in some sense. So now I want to end by showing you a trick, so to speak, that we developed now using AI. So now we have neurons from human or from mouse. We have many details, anatomy, location of synapses, properties of inputs. We have many knowledge about the cells, and we can build a mathematical model of the cell in the computer. So there is a mathematical replica of the cell in the computer, and I can ask questions about how this cell will behave to this input, to this current, to this synapse, to this, and I can explore the input, output properties of this particular cell. This is what you see here. It's a mathematical modeling of a particular cell that we took in this case from a mouse and we studied. Recently, these two students came with an idea, together with Nicky London, who is part of ELSEC, and a colleague of mine, that we can look at this input-output of this biophysical cell and make a replica, and we call it twin. We make it a deep neural network twin. You may know that deep neural networks that we developed and I mentioned before are very good in replicating input-output properties of anything, and in this example of a cell. So we can ask a question, how deep should the artificial neural network be in order to replicate one by one all the inputs that I gave here to this input layer of the deep neural network AI. So I'm building now a twin model, mathematical twin model, but using this technology of the AI in order to replicate the biophysical model that I can do in the computer. This is this new technology that we developed in the lab, and this enables us to ask questions directly. Are human neurons deeper, more clever, because when you have a deeper network, you are likely to be able to solve more difficult questions or issues relative to a shallow network. So this is a very recent result, and here is the idea. You challenge the human neuron, the model of the human neuron, you want to challenge with the question, can you teach this neuron to characterize, to classify between dogs and cats? Yes, I want this neuron to give me an output only for all the dogs that I show him as input, and zero, no output, let's say, for cats. But I don't know how to do it to the biophysical model. I don't know how to teach the biophysical model to distinguish between cats and dogs. But I know how to do it in deep neural networks, because this is exactly what deep neural networks know to do. You use certain algorithm to give an input and to teach the network to give an output only for dogs and not for cats. So because I have a replica of a neuron into a deep neural network, and I can ask, can I teach this deep neural network, which is a replica of my neuron, to characterize between cat and dog? And the answer is that yes, I can take a human neuron, build a replica that is identical, twin to it, and this replica can characterize between dogs and cats. But if I take a mouse neuron, which is smaller, less connection, and I build a replica and deep neural network replica of this biophysical neuron, then it cannot solve the problem. So already at the level of a single neuron, our microchips are more sophisticated than the microchips of the mouse. And of course, this is not what makes us human. What makes us human are many, 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 many little things that came together. Already at the synapse and at the microchip level, we are also more sophisticated, and we have more elements, and we have more connections, and we have more regions, and eventually we come to the brain circle, and the mouse does not. Maybe there is a mouse here. Okay, so I want to very fast finish with three things that I think are going to change the world. What I already told you is going to change the world, and then I'm done. I want to show you this thing that everybody knows. This is a Big Jump 2022, amazing women. Jennifer Doudna and Emmanuel Champartier. They got Nobel Prize for the CRISPR technology. The CRISPR technology is a technology that enables you to edit genes. You can cut a gene, a DNA at a given location, and put inside this particular location another DNA, human or not human. So this is a really complete change of technology, I think it's a big game changer, because we can now manipulate the genetic itself, we can change the DNA itself, we can generate new species by genetics and so forth. I'm not going to talk about that. But there are two other things that again I learned from you, because I needed to give this talk. Recently, using genetic technology, people succeeded to do the following. You can take a mouse male, you can take a cell from a mouse male, you can by genetics take the genome of this single cell and make it an egg cell. Okay, so this is a mouse cell. You can manipulate the genetics of this mouse cell, you reverse engineer it to become stem cell. So this is a grown mouse, you make it a stem cell, as I showed before, but from the male cell, you make it a female cell, an egg. Then you can take a sperm from a mouse, another mouse, and make a new mouse from two male mouse. And this is already happening. So these puppets, you see these puppets, are the result of two males. One was manipulated genetically to become an egg cell, another one was not. And eventually,
There is a question if
Would it work if it's the same, or they need to mix DNA? If it's the same male, but not incest, like himself, basically.
no, but these are two
I
males.
understand, but would it work if it's the same?
Or from the same male to do from me to do an egg.
me to me.
Me to me.
You take an egg.
This was not tested. I don't know.
It would probably work, but it would be
It
like
should,
incest.
but this I don't know. It
It
should.
would be immoral.
It should.
No, I would mean that the mouse would have probably defaults because it doesn't have enough genetic variety.
Exactly.
Right, yes. But in this case, so this is a creation of a mammal. This already happened. So there are these males, puppies that are a result of two males. But this is a creation of a mammal with two biological fathers. This could be paved the way. There are many gay friends of mine that are very happy from this because now they can make between them kids. That's one big jump. But I think the most dramatic jump, and that's part of what you asked before, is really the fact that we can now develop not only the genetic, but also there are a lot of attempts in the world to develop artificial womb. Where you don't need to be any more pregnant, so to speak, within yourself, but you can do it outside after fertilization. And this is already starting in several labs in the world. People have already succeeded also in Weizmann Institute. Jakub Hanna and others, they could already take outside and develop artificial womb and enable it to grow, not to the level of birth, but to the level that you can start to grow tissues that you ask for. So we can now grow pieces of heart and pieces of heart in this technology. I must tell you, and by this I want to end, in the last 3 years I had a course with Idan Segev about the future of technology and the future of humanity. And when I showed this to the students in the lab, in the group, there were 15 male and 50 female from different disciplines, I asked them, female, what would you say if there would be an artificial womb, completely secure, safe, everything is fine, and you know that you can have a partner, male, female, whoever you want, and make it outside. Will you choose to make your kid outside or inside? This for the first time. I didn't know. I thought I would know what would be the answer, but I let them answer. 15. One of them said certainly at least once inside. The other 14 said not at all. Why should I do it inside? With my body, it should be outside. Then I realized that I'm a little bit out of phase of the young generation. I was shocked by the answer they said. I thought of myself as being young forever, but they think differently than me. So this is a change. This is a really, really game-changing technology. Finally, and I do it every time. Finally, I cannot avoid telling the young generation here and the parents of the young generation here that we need to prepare this generation to what I just told you about, because the world in 20 years is not certain. The brain center will continue, but the world will look different. It's not going to be the same world. There will be AI, there will be robot, there will be avatar, there will be genetic manipulation, there will be brain-machine interface, whatever we're talking about. It's not going to be the same world. We have to prepare these kids. We have to prepare these kids. So that's what we are trying to do in Frontiers for Young Minds. If you take this, or just log into, then you will see many of the papers related to what I spoke about today are already written for kids for the next generation. So they will be able, in a nice way, in a simple way, to understand where we are headed. I want to thank you very much. Yes.
Going back to organoids, I'm assuming they don't develop functional specification yet when they're that small. How long do you think it would take you?
What do you mean functional benefits?
They don't become frontal cortex or
They do. You can manipulate them early on to become visual cortex, somatosensory cortex, thalamus, other regions. You can force them, you can manipulate them, and
But only once implemented into a chimera, or
no,
even on their own?
outside they develop the cell types and so on that are particular to this region. Yes, because we know how to reverse engineer and then manipulate it with certain environment and certain manipulation to become a cortex, a thalamus, a stem cell, a spinal cord. Outside. That's how they did this connector. They did it outside piece of the spinal cord, outside piece of the cortex.
So it's not
And
just
then they connected them outside, but
Right.
only outside had the property of the sensory system and the spinal cord system and so on, outside.
it's not just a plot of neurons.
It's not
It's
plot of
already
neurons.
developing its own dendrites and
Yes.
its own.
Part of the eye, the retina. Yes, they develop specificity if you want to diverge it. As in the brain, eventually you start with the same cell, two cells become eventually all of you. So inside the body they become specialized to this part because it's the same genome. But you can do it also on the outside. Not to this level of specificity. Yes, I'm done. Thank you very much again.
First of all, I really want to thank you for this lecture. You didn't say the mouse started to change its behavior. I didn't hear that. You said you would say that, but then you avoided. And I think it would be interesting. I want to really to know what's happening. It's a big question. Sorry for the interruption. I have to go to the US Embassy because the Minister of Interior of the United States is there and I have to meet him. But, Your Excellency, thank you very much for this invitation. There are many reasons I admire Israel, and I stand with Israel all the way in all this difficult period, and publicly, and very loudly. One of the reasons is the level of research, development, and science you bring as a gift to humankind. And this is totally marvelous. Professor, this was outstanding. As a Minister of Health, I couldn't stand thinking the budget cost of all these for me, but okay. From the other hand, everybody can understand that this could lead to new treatments that today we cannot even imagine, and diseases that really makes life terrible, like Parkinson, for example, or other brain diseases that maybe with this technology could be reversed. And this would be a miracle for humankind if we think that we are an aging population, not you in Israel, but you in the rest of the West. And with age, all these diseases come more. So we'll need much more of this technology in the future to come. So thank you very much for the invitation. You are very welcome to our country, extremely welcome to our country. We are in a totally friendly country, and we are very proud that you are our friends. Thank you very much.
The minister wants to know if the rat will respond, will start to talk.
Yeah, not talk, but change the behavior.
In change of the behavior. So we have to, because as I said, because the human organoids in the rat is now about 3 or 4. A year boy is not enough. You have to wait for it to grow. Then within a year, invite me to Greece. And I'll give you
Have an open invitation with a mouse. Please, this is really important.
Okay, this is a mouse.
The next time.